So I have to admit, well, thank you for the invitation. And I have to admit, yesterday when I walked in, I thought I was at the wrong meeting. Now, I love the brain as much as the next person. Do you want to figure out why the slides aren't coming up? In fact, both my parents were psychotherapists, and I think I was their main experimental target, violating every rule, but that was when it was still okay. So some of you can empathize with me. There are whole books written about me, right? And I was also a child of Silicon Valley. My father was the therapist to the founders of Silicon Valley. And by the way, I have some great stories for you if you want to hear them over lunch. Growing up there in that time was amazing. But I think because of that, I also was just inundated with science, math, and engineering. I went to, the kid who lived across the street and I went to elementary school with actually invented the robotic arm when he was at MIT. So there was so I grew up in this very interesting environment, and I think it propelled me to think about engineering, and also simultaneously during my growth as a researcher, the advent of molecular biology was happening. And so synergizing all of those things and thinking about how we can use biology to do good things. And that brings me to what I think, so I'm not going to talk about any brain stuff except what's in my own brain, but it brings me to a question that I think we all should be struggling with, and that especially the next generation of scientists and engineers is, how the heck are we going to make a world that can support twice as many people? And that extends to all aspects of life and behavior. Oh, and by the way, I wanted to add one more thing about what I've been hearing so far at this meeting, is a lot of the language you use about ethics and DIY and all of those things resonate for me just as much, because in the engineering of biology and what we do, all of these same issues apply. So I actually celebrate the conversations around those topics, and they're highly applicable to what we do as well. But what are we going to do here? How are we going to fix this problem? And I believe that we can fix it through the engineering of biology. We have a deep knowledge that allows us to begin to think about how we can engineer all aspects of life, whether it's bacteria, plants, mammalian cells, and we can engineer their interface with humans, or mammals, which is one of the things I work on. But we also can use them to engineer our interface with the environment. Now, some of you are old enough to recall where this picture comes from, maybe? This is the Earthrise, right? So this was the front page, or the cover of the Whole Earth catalog, which almost nobody remembers here, but was inspirational for me. It was kind of the pre-Amazon, right? It also articulates another thing that I think has captured the interest of young people, be it far-fetched, is the prospect of going to Mars. For me, it was the moon when I was growing up, and that turned out to be successful. It's not clear. We got a lot of technology out of it. We haven't done much else with it. But the idea of sustainability on Mars has really captured the imagination of a lot of young people. And so those two, of course, integrate. How do we sustain Earth, as well as how do we generate a community on Mars? So how did I get into this? So prior to, so I had another life as a biologist, molecular biologist, studying basic fundamental problems in biology. I then went on to work at Dana-Farber Cancer Institute. I actually developed a drug that is, I'm pleased to say, is now getting FDA approval for cancer, so I feel really good about that. But at the same time, I felt like, okay, we have this vast knowledge of how biology works. How can we start to use that to do things with? And I had the good fortune, as I said, to be in Boston and to meet a group of bioengineers and computer scientists at MIT. Jerry Sussman was one of them, Drew Endy, Tom Knight. And they welcomed me into their group as sort of the token biologist. And they asked the question, coming from their point of view of internet and computer chip design, et cetera, why can't the engineering of biology be faster, cheaper, and more predictable? And they asked that question. We began the synthetic, we called ourselves the Synthetic Biology Working Group. Starting in 2002, we actually issued a ISAT report that was funded by DARPA for a roadmap of how we could achieve this. Unfortunately, you may remember, it's also coincided with John Poindexter being fired, and he was running the TIA program at DARPA. It was the one time that DARPA, I think, lost their funding. And so TIA is the Total Information Awareness Program, which, you know, Ed Snowden got in trouble for. It still exists, but it's just not run out of DARPA anymore, right? And so we had the misfortune of, our timing was bad, but now it's okay. But you can actually read this report on the internet if you want, and it lays out some of the things that we felt were necessary to make this happen. And I'm gratified to say that we have achieved most of the goals of that roadmap. And so we are now on to making the next roadmap for synthetic biology. Now, as I said, for me, there are two big desires here. One is the engineering of sustainability, which I will talk about, and is why I'm invited to this meeting, is how do we convert sunlight into stuff, which is what plants do, and so how do we mimic what plants do, and hence the interface with inorganics and life. But we also have a huge interest in health and developing new kinds of therapeutics. I actually have an idea for something to do with traumatic brain injury, which I'd be happy to talk about. And also, I think I don't even almost have to say this anymore, that the future economy, at least where I live, is really based around engineering biology. And you can call this a green science or a green technology. And a lot of what we do is incentivized by engineering biology so that we can avoid use of petroleum products. Okay, so I'm going to talk about one thing, which is a device we've developed whereby we can harvest light and send it to a biological system, to bacteria that are highly programmable, and I'm going to say in a big sense, could almost be programmed to make anything you want. So sunlight is our greatest natural resource, more so for you guys than for me, maybe. And we use a very small proportion. We use a very small portion of it. I gotta say, I just want to tell one anecdote about. I was at a meeting, an energy meeting, and there was a guy passing through on his way to. Sorry, this just stays on. A guy on his way through to begins installing solar panels across all of Saudi Arabia. And as some of you may know, they're thinking about that kind of future. Of course, that isn't going to go so well with our interaction with them right now. Now, about energy in general. So we're going to have a lot of new energy users as the world population grows. A lot of those people are going to be in the developing world. Now, in the developed world, we already have a style of energy usage which is centralized and again is based around the petroleum industry. But we'd like to imagine that a future energy structure could be more distributed, localized, more available to all, and have a low capital expenditure. And there are some great analyses of using solar power and how we may almost be there economically, but a lot of the problems are social and political. Now, there already is a technology that does this really well. It's called a leaf. And so a lot of what we do is try to imitate what nature already does. And so photosynthesis, this is an oversimplification, does something called the water splitting reaction in response to light. And then this, again, an oversimplification, but this can result then in the fixation of CO2 and the accumulation of biomass. So this is a technology that works really well. This is what the planet sustains the planet. So we would like to think about how we can harness this technology and focus it into processes that could be beneficial. Now, the story I'm going to tell is also a case of serendipity. So one thing some of you may know at Harvard, we almost never talk, the faculty almost never talk to each other, right? We have what I call acquired situational narcissism, which is a term I developed. So you could, every time I tell psychiatrists this, they've never heard that term. And so, right, so we now, in a rare happenstance, I was working on, so I had this dream about making biohydrogen in response to light. So I was trying to engineer light harvesting bacteria to make hydrogen. It probably, I think Carolyn was in the lab then, it probably violated the laws of physics, but it sounded cool. Meanwhile, I met a colleague in the chemistry department, Dan Nocera, and I literally met him at a holiday cocktail party. So like I said, the only time that Harvard faculty almost ever talk to each other. And fortunately, Dan said, well, I've been really wanting to meet you because as a chemist, he had developed something called the artificial leaf. Now, his desire was, in fact, to create a totally artificial photosynthesis reaction whereby light could be harvested, do the water splitting reaction, and generate hydrogen. And that is his invention, which is really key to what I'm going to talk about. And this is the electrocatalyst, which is based on the original version, based on a cobalt phosphate and nickel molybdenum zinc catalyst. I realize a lot of you aren't chemists, but in response to light, would activate this electrocatalyst to do the water splitting reaction and make hydrogen. I only want to show this because over here on the right is the actual artificial leaf, and hopefully it's kind of light. I don't know. Oh, there you can see it. This is actually sitting in a jar and it's shining light on it, and those bubbles are hydrogen coming off. By the way, this makes great substrate for elementary school presentations. Dan and I go around, and we did one to first graders. It's amazing how they can get stuff, especially if it's visual. Sorry, you can't really see that very well. But anyway, so this is a thing that's making hydrogen. Now, Dan and I both sort of got trapped in the Stephen Chu world of there wasn't going to be a hydrogen economy. Remember that? And it was all going to be about electric cars. And so the vision of hydrogen, let's just say it wasn't very popular. And so Dan was really looking for a way so he really had a solution of producing hydrogen, but a lot of the problems around you can't store hydrogen, again, localized production, et cetera, led him to wonder, is there an alternative approach whereby we could use biology to essentially store the hydrogen? And there is, and that's where we came in. Because by growing certain kinds of bacteria together with the artificial leaf, whereby the bacteria can use the hydrogen and fix CO2 and then make stuff. These bacteria are called Ralstonia. There are other types which I will talk about towards the end. But this is the basic idea, and this is what we invented together. So what we're doing here is taking photosynthesis, taking a bacteria that can use hydrogen to drive the Calvin cycle, and then providing the hydrogen via an inorganic man-made system. So that's the basic idea. This is what it looks like. We call it the bionic leaf. Reporters come and say, well, what does it look like? And it actually just looks like a first grade science or a sixth grade science experiment. It's literally 2 electrodes in a jar sitting on the bench. The thing that's unique about this system is the bacteria can grow together with the electrodes. That's one key development that we discovered. Because in the first version, that one I showed you of Dan's original artificial leaf, it worked. The cells would grow, they would make stuff, but not very well. And this is a chemistry fact that particular electrode pair was generating things that were toxic to the cells. And this is a problem that you often run into when you're trying to interface inorganics with life. And so the other thing that Dan's lab invented was a new kind of electrode pair based on cobalt and phosphate, whereby it is no longer toxic, it doesn't generate reactive oxygen species, and it also does something that's called self-healing. So as the cobalt comes off, it can actually redeposit on the electrode. So the beauty of that is that this thing can keep running for a long time. You don't have to keep refreshing it. So we called it the bionic leaf. We showed that it can produce biomass. We can scale it up to a benchtop scale of 1 liter. We build a 5-liter scale up. So those of you can pester me about how the heck this is ever going to be scaled up. I'm not a chemical engineer, but there are people working on that. And there's a story behind that. Now, the other thing that we're interested in is the back end. What can you get this system to actually produce? And the bacteria themselves actually produce polyhydroxybutyrate, or PHB, which is a plastic precursor. And so in our system, they're also accumulating PHB, and we can also program them through genetic engineering to make other kinds of molecules, such as different kinds of alcohols, as an example. So somebody may tell me this is out of date, but at the time that we developed this, if we looked at the yield based on the theoretical incoming from solar, this would, in terms of biomass accumulation and CO2 fixation, the efficiency would be around 10%. As some of you may know, plants are the least efficient, which is interesting to think about, down there at 1%. Algae are sort of the gold standard at 8%. So as of when we published this was sort of the gold standard. So this is a list of some of the things we can make, and I just want to focus on glucose. So glucose is a feedstock, and this project actually stemmed from a grant that we wrote for NASA, which, by the way, we didn't get, but I'm told that's no big surprise. But the idea of, can we now produce a feedstock that could then go and feed other things to grow? And glucose is a key feedstock. So we want to alter the bionic leaf to produce a feedstock that then can be co-cultured with other organisms to make yet more things. So sorry about the chemistry here. The bacteria themselves are doing the Calvin cycle. Hopefully they still taught this to you. They still teach this here at Rice. I think we teach it at Harvard still. And there are 2 enzymes that you need to add back to get the cells to make glucose. So to make a very long story short, we can add back those enzymes, and we can measure sucrose production. Sorry, I meant to say sucrose. Now, there's a problem. which is the sucrose. So in many types of cells, they will actually push the sucrose out, but not in these. So it's trapped within the cell. So we have another trick that as engineers we can do, which is to insert special proteins into the membranes of the cells that kind of act as tunnels that will allow the sucrose to be excreted. And that too was successful, whereby if you just look at these dots, these are the level of sucrose that's actually being found in the supernatant from the cells. So you get rid of the cells and you measure how much is left in the supernatant, the soluble phase, and that's where all the sucrose is going. Moreover, there's no inhibition of making sucrose for the cells. And finally, the sucrose can be co-cultured together with other kinds of cells, like yeast. So yeast is a major industrial organism. It's more than just making beer. You can actually make opiates with yeast, for example. So this would be an autonomous system whereby it would produce the feedstock that can drive through sunlight or some external power source that could then feed into an industry such as growing yeast or bacteria where, for example, vitamins come from. Right. Okay, so these are some of the features of the system. It takes up less land mass. It's modular, so the system could live in your basement and you could harvest energy on your roof, and so the two don't have to be in direct contact like algae does. So there was some discussion last night. I want to finish with this, two things. One is there's another big problem facing the world, and that is fertilizer. So when, early last century, the production of ammonia escalated, so did the production of feed crops, and this caused, of course, just essentially a revolution in life on Earth. But the production of ammonia is extremely costly to the environment. It produces a huge amount of CO2, estimated amazingly to be at 1 to 2% of, I think, the CO2 that gets generated. It's a high energy, high intensive, high temperature process. On the other hand, their nature can fix nitrogen. There are microbes that are capable of harvesting nitrogen, and this is one of them that we have studied because this microbe, a form of xanthobacter, can actually fix CO2. It can use hydrogen, and it accumulates it in these large granules that, again, have PHB, but can also produce, it can fix nitrogen and hence be beneficial to plants. So what we showed is that you could take these xanthobacter, because remember, they fix nitrogen, which is a good thing. They fix CO2, which is a good thing, and they use hydrogen. And so we can grow them here in the bionic leaf setup, just like the other bacteria. And I'm not going to belabor this. They actually grow, and they actually make ammonia. We can do that chemically. And I want to show this last slide, second or almost last slide, whereby we can take these bacteria, grow them in the bionic leaf, and I discovered that at Harvard there's something called the Arboretum, which is a beautiful place, but they also allow you to do experiments with genetically modified organisms on plants. And so we were able to test these bacteria on their impact of growth of, go figure, radishes. I think that's a popular space food. And indeed, here are radishes with no bacteria or a low level up to radishes that have been supplemented in the soil with our bionic leaf-grown bacteria, and hopefully you can see that they are significantly larger. So it works as a fertilizer. So I want to close with a little bit of something that is on a lot of people's minds is, okay, we can do all this stuff with organisms. There's a huge opportunity here, but there's also a perceived risk, which is not just perceived, of how are we going to release things in the environment. My feeling is that we have to sustain the Earth, but we also have to confront how we're going to do that in a safe way. And so what are the things that we worry about? So one is contamination of the environment, and there are solutions to that problem whereby you can build in ways for the cells to commit suicide. These are called kill switches, or in computer world, dead man switches. Someone pointed out it's a bad optic, we should probably have a better name. But the one I want to just close with is this idea of genome recoding. Because if you can change the genome so that it does not look like the version that lives in the environment, then you can prohibit what we should be worried about, which is horizontal gene transfer, which is something that happens in nature and drives a lot of evolutionary processes. So how do we do that? Now, we take advantage of the conservation of the genetic code. With possible few exceptions, it's conserved across all of life. There's a redundancy in the genetic code in that you have multiple codons for certain amino acids, such as leucine, arginine, and serine. And what this means is that you can change the genetic code such that you can get rid of, say, 2 of those serine codons and get rid of the corresponding tRNA. So those cells then are incapable of supporting foreign DNA. And if you don't understand that, I can explain it later. This also has a huge economic advantage because bacteriophage and viruses will be unable to grow, and that has a huge economic value. So I want to end with saying that the potential for what we could put into the environment, I think, has huge opportunities. As I said, one of the things we work on is engineering the gut microbiome for medicine, but there's opportunities in bioremediation, detection. The list is endless, what you can do with biology. And if you run out of ideas, I run the iGEM competition. Rice has an amazing team, by the way. And if you ever run out of ideas, just look online each year at the Jamboree. There are so many great ideas there, and most of them are not being implemented because the students go off and do other things, which is good, but also because of this prohibition about how we're going to release these things and do all these wonderful things. So this is really crippling application development. But now, because again, going back to that DARPA roadmap, one of the things we achieved was the cheaper, faster, better synthesis of DNA, which is the substrate for all of what I've told you. So now it is financially possible to actually resynthesize the genome of a bacteria. And so under $1 million, that price will still continue to drop. So this is a reality. You could say that going forward, perhaps all engineering in laboratories should start with a recoded organism. And then I'll end with Dan and I had this vision of, sort of DIY homebrew energy, or at least localized as opposed to distributed, whereby you could harvest energy and direct it into fuels, materials, drugs, and food. All of those things we need for that future Earth. And these are my colleagues, these are my conflicts of interest, and these are my funding. So thank you very much.
Thank you. We have time for questions. I remember Frank Dyson when he was explaining why he was a technological optimist and not so worried about climate change. He talked about forests of artificial trees. So how about scaling up your leads?
Yeah. So scale up. As I said, I'm not a chemical engineer, but I will tell you a kind of a cool story. I'll make it brief. So Dan has been working in this field a lot longer than I have, but I did venture into the private sector a little bit. And one thing that you probably know is funding in alternative energies oscillates, and it particularly oscillates with the election cycle, I think. And many times every president comes in and says, we're going to do alternative energy, except the current one. And so there's a huge oscillation to funding. It is also not compatible. I'm going to answer your question. It's not compatible with the current venture funding strategy, which is short term. Now, if you look at the developing world and the members of the Paris Accords, they have financial incentive to develop new technologies. So we have partners in India where they have a huge incentive to explore how to scale up this technology. Frankly, I don't have a clue how to do it. But, and distribute, I like your idea of, I should say, your idea of little tiny farms, though, having these things is one of our commercial ventures as well.
Yeah, so I wanted to ask, in these bionic leaf devices, so you have the silicon multi-layer device, and that absorbs the sunlight, then you have the bacteria in the solution or near that device. Do you have to do something to engineer that interface, or to get the bacteria close, or to get it kind of well mixed? Right. And how do you, what kind of things do you need to do, or is it not a big barrier? Yeah, well, I'm curious about that interface.
Historically, I mean, other people have done similar things, and what you're referring to is historically there's a barrier put up between the device and the living part because of that very problem. And what I perhaps didn't make clear is that we sort of had that problem with version 1.0, whereby reactive oxygen species would be generated. The solution, which I think was amazing, was what Dan did, was to invent around it and to find a electrode pair that was not toxic, didn't generate reactive oxygen species. So that's what makes our system different, is that we can grow them directly.
I have a suggestion and a question. The suggestion is, title something you write, "Do Androids Dream of Electric Leaves?"
I love it.
And then somebody will change the name and turn it into a really big blockbuster movie.
Actually, there is a best movie. I did a movie review of this movie, Annihilation. Best movie about horizontal gene transfer.
That actually was my question. So I think it's the idea of eliminating some of the homonyms is clever, and the tRNAs for the homonyms is clever. But that prevents transfer in. Doesn't seem to prevent transfer out.
So smart. Now, I would give away my secret, but I will. You can, because what you can do is, so you are correct, the incoming has a problem, and that's a big deal. The outgoing, this is the concept, is to have them and have genes in them that would normally be toxic, but because they have the complete genetic code, won't be translatable until they are transferred out. So that's the trick.
Okay.
You're supposed to love that idea.
I need to think about it.
Don't show so much enthusiasm.
But fascinating work, and I hadn't heard of it before. Thank you.
So of course, I'm really excited about the environmental synthetic biology you're doing. You kind of alluded to this idea of scalability being an after thing. And I actually feel like the field right now isn't really thinking about scalability. And so I guess I pose the question, should we be really thinking about this on the front end? I mean, the problems you're talking about in the time scale feel very short to me, and we need solutions. And so I guess, I'd push back on that of not leaving it to the chemical engineers, but what should we be doing?
So I actually probably worded that incorrectly. As a trained laboratory scientist, I do, much like many of my colleagues, I do tend to do things at a benchtop scale, because that's pretty much all I know how to do. However, most people in industry would tell you that what we do at the benchtop never translates to industry, and and they always have to rejigger it. So, what I what I propose is just that there should be more communication between us and the people. And there are efforts to do that through various companies are very interested in collaborations in that space. And actually, I think it should become a whole curriculum that is integrated with synthetic biology. And I say this, and then somebody said, well, we had that at MIT. And so apparently it did exist at one time. But I think it should become more of an integral part of a synthetic biology curriculum because most people are working totally in isolation.
So most bacteria have evolved various ways of getting sucrose inside their cells because it's a great source of energy. How stable are these constructs where you're exporting energy on an evolutionary timescale?
So I'll begin with the cyanobacteria story where we did this first. So we engineered cyanobacteria which can harvest light directly, and we engineered them to make sucrose. And the problem was, how do you get it out? And we actually used the sucrose antiporter because, interestingly, in that case, the bacteria grow in high pH, so you can reverse the direction so that the sucrose goes out. So we thought that was really cool and super stable. Actually, people are using those in scale-up reactors, which I was quite excited about. They still suffer from the problem of they have to be exposed to light. In this particular case, we're using a passive one, so we didn't do the pH trick. We're going to try it. And so far, so secreting the sucrose actually, this was buried in the slide, actually gives the cells a growth advantage. Because if they're busy cycling carbon into sucrose, that's not so happy unless they can get rid of it and thereby change the flux through the pathway. And so it's a flux balance situation whereby it actually, they do better. Go figure. We actually, yeah. Okay. Thank you.
Thank you.